This is Chapter 24, Module 5, Digestion and Absorption of Carbohydrates, Lipids, and Proteins. The learning objectives of this module are 1. Describe the chemical events responsible for the digestion of carbohydrates, lipids, and proteins. And number 2, describe the mechanisms involved in the absorption of carbohydrates, lipids, and proteins.
We'll start with the life of a carbohydrate. Keep in mind that a complex carbohydrate is called a polysaccharide. This must be broken down into small complexes called disaccharides and trisaccharides, which must then be broken down into the smallest unit called a monosaccharide. It is the monosaccharide that will be absorbed. Carbohydrates begin their chemical digestion in the mouth. The salivary amylase secreted by the salivary glands will begin the breakdown of carbohydrates. For the most part, no absorption of carbohydrates takes place in the mouth. The carbohydrates enter the esophagus where the salivary amylase will continue to chemically digest the carbohydrates. These polysaccharides will be broken down into disaccharides and trisaccharides by these enzymes. The disaccharides are carried to the stomach. In the stomach, after about 1 to 2 hours, the hydrochloric acid drops the pH to 2.0 or below, and this stops the activity of the salivary amylase. So chemical digestion of the remaining polysaccharides stops temporarily in the stomach. In the duodenum, pancreatic amylase is released into the duodenum and will break down the remaining complex carbohydrates. Any disaccharides and trisaccharides produced will not be broken down any further until they contact the intestinal mucosa. Brush border enzymes of the intestinal microvilli break these disaccharides and trisaccharides into monosaccharides, or simple sugars, that are then small enough to be absorbed. The intestinal epithelium then absorbs the monosaccharides by facilitated diffusion and cotransport mechanisms. The simple sugars that are transported into the cell will diffuse into the cytoplasm of the epithelial cells, then carried back by facilitated diffusion across the basement membrane. From there, the monosaccharides will diffuse into the capillaries of the villi, where they will be transported to the liver through blood vessels of the hepatic portal system. Now it's your turn. Draw a concept map of a carbohydrate as it begins as a polysaccharide and ends up as a monosaccharide that diffuses into the bloodstream and transported to the liver. Next, we'll look at the life of a lipid. Keep in mind that the lipid is usually ingested as a triglyceride. The triglycerides congregate together as a large lipid drop. Triglycerides get broken down into monoglycerides and fatty acids. Lipids begin their chemical digestion in the mouth. The most important and abundant dietary lipids are the triglycerides. These triglycerides tend to form large lipid drops as their hydrophobic ends repel away from water. These lipid drops are large enough that only about 20% of them will be digested by the time they enter the duodenum. Small amounts of lingual lipase from the small glands of the tongue begin breaking down these triglycerides that are ingested. 2 fatty acid tails are broken off the glycerol head, leaving 1 fatty acid tail and a glycerol, which we now call a monoglyceride. Bile salts from the liver and the gallbladder will help us to emulsify these large drops into tiny droplets, making it easier for further breakdown. This emulsification occurs only after the chyme has been mixed with the bile in the duodenum. Pancreatic lipase released from the pancreas will enter the duodenum and break down the tiny droplets of triglycerides into a mixture of fatty acids and monoglycerides. These fatty acids and monoglycerides will react with bile salts to form micelles. When a micelle contacts the intestinal epithelium, the lipids diffuse across the cell membrane of the epithelium and enter the cytoplasm. The intestinal cell walls synthesize new triglycerides from these monoglycerides and fatty acids. These new triglycerides are then coated with proteins, and the resulting complexes are called chylomicrons. The protein coating keeps the chylomicrons suspended in the interstitial fluid, but they are too large to enter into the blood capillaries. Instead, they diffuse into the intestinal lacteals, and from there they are transported through larger lymphatic vessels toward the heart. Close to the heart, the lymphatic vessels deposit their fluid into the subclavian veins, and the lymphatic fluid, along with the chylomicrons, are deposited into the blood. Most of the chylomicrons and bile salts are reabsorbed by the body. Only about 1% of the bile salts leave the body in the feces. Now it's your turn. Draw a concept map of a lipid as it begins as triglycerides bundled together in a large lipid, then breaks down into monoglycerides and fatty acids, then reacts with bile salts to form a micelle, then diffuses across the cell membrane where new triglycerides are formed from the monoglycerides and fatty acids, then coated with proteins, and finally then diffused into the lacteals where they will be carried close to the heart and deposited into the subclavian veins and mix with blood. Next we're going to talk about the life of a protein. Proteins are complex, so protein digestion is complex and time-consuming. First, mechanical breakdown by the teeth occurs in the mouth. Then chemical processing begins in the stomach. There's no chemical digestion of proteins in the mouth. The strong acid in the stomach kills pathogens and breaks down plant cell walls and animal connective tissues. This acid also disrupts the tertiary and secondary protein structures, exposing the peptide bonds. The acidic contents of the stomach also help increase the activity of pepsin, which is secreted by the chief cells of the stomach. This is a proteolytic enzyme. In other words, it breaks the peptide bonds within the proteins, or within the polypeptides. This enzyme works well at the low pH of the stomach, which is 1.5 to 2.0. When the chyme reaches the duodenum, the proteases from the pancreas can begin working. These proteases are trypsin, chymotrypsin, and elastase. They act like pepsin and break specific peptide bonds within a polypeptide, creating shorter polypeptide chains. The epithelial surface of the small intestine contains several peptidases, most notably dipeptidases. These are enzymes that will break down short polypeptide chains into individual amino acids. These amino acids are absorbed through facilitated diffusion and co-transport mechanisms into the blood capillaries found within the villi. The amino acids enter the blood, which will then be transported to the liver through the hepatic portal system. Now it's your turn. Draw a concept map of how a large protein, or a polypeptide, is mechanically broken down in the mouth, enters the stomach to be broken down into a shorter polypeptide chain by the acid and the pepsin, enters the duodenum to be further broken down into small peptide chains and then broken apart by dipeptidases into individual amino acids, and finally transported into the blood capillaries to be transported to the liver.
This ends Chapter 24, Module 5, Digestion and Absorption of Carbohydrates, Lipids, and Proteins.